So when Francis Crick proposed DNA makes RNA makes protein, we have to understand how a DNA containing just 4 bases, like G, A, T, C, can make RNA, which also contain 4 bases, G, A, U, and C, and proteins contain 20 different amino acids. So there is an inherent complexity as to how this whole transcription and translation is going to function. But even before getting into that, we need to understand what is gene expression. So expression of a gene, we understood from the previous chapter that genes are made up of DNA. So expressing DNA is that taking the information from DNA and then converting into some other message so that that message can be used to make protein. Okay? So in this context, DNA is that information, genetic information, RNA is the message, and protein is the one that would perform different functions. So the expression of gene refers to either synthesis of RNA at the transcriptional level or synthesis of protein at the translational level. So when we look at expression of gene at the transcriptional level, you need to understand what genes are made up of. What defines a gene? So gene is a fragment of DNA of specific sequence that contains a coding information to make just RNA and protein. Because not all RNA are converted into protein, like ribosomal RNA, tRNA, they do not make protein. Whereas the mRNA contains information to make protein. So in this context, when you think of a gene, it contains a coding region to make this RNA, and there should be a control element on one side of the gene, it's normally called as upstream of the gene, that can regulate this gene expression. So a functioning gene will have the control element called as a promoter, the coding region that contains information to make the RNA, ribosomal RNA, tRNA, or mRNA, that set is considered as a gene. So a coding sequence alone without the promoter is considered as a pseudogene. Okay? So that's not a true gene or expressing functional gene. So once we understood this part, the next thing to understand, how you're going to make the RNA. What are the players involved in this RNA? If you look at that, there are 2 major players involved. One is the enzyme that makes RNA, called RNA polymerase. Like the enzyme that made DNA is called as DNA polymerase. The enzyme that makes RNA is RNA polymerase. And there are proteins that regulate transcription called as transcription factors, or also known as transacting factors. So these transcription factors bind to the promoter region, then RNA polymerase comes and bind to that. Then they start to open up the DNA and they start to make RNA from the DNA. So the RNA is made from 5' to 3' as a new strand on a template strand that runs in the opposite direction, 3' to 5'. So understand how this process works. What is a promoter? A specific set of sequence on the DNA recognized by transcription factors, and that also binds to RNA polymerase. They open up the DNA, start to read the template strand and make the RNA. Once the RNA is made in prokaryotes, it's very simple. RNA is made and translated, or proteins are made simultaneously in the same place, but eukaryotes, it's a little bit more complex. The RNA synthesis happens in nucleus, and then it moves on to processing of RNA. RNA processing involves addition of a G cap at one end and poly A tail at the other end. So the G cap is added on the 5' end of RNA to protect the RNA. The same at the 3' you have long poly A tail, which does not form any specific secondary structure. It's also used for stabilizing RNA or protecting RNA from degradation. So once this processing is done, there's one more step in RNA processing, that is to remove intervening regions called introns in eukaryotes and then connecting exons. So this is called as RNA splicing. So this also happened in the nucleus. So just to review what we have seen, the gene expression is expressing the information stored in DNA in the form of RNA, And that happens when transcription factors bind to the promoter, open up the DNA, make the RNA. Then in eukaryotes, the RNA processing happens by adding a G cap and poly A tail and removing introns and adding exons. Also, one more thing to remember is the G cap and poly A tail are only for mRNA, and the introns, exons could be present in either mRNA, tRNA, or ribosomal RNA. Once the processing is done, the RNA leaves the nucleus. It starts the process of protein synthesis. So we have to also understand, before you step into protein synthesis, what is the principle behind protein synthesis? What is going to happen there? So if you think of the logic, the mRNA contains the coding information called as codons, and they can be only of 4 bases, G, A, U, and C. How these 4 bases can code for 20 different amino acids? So that itself is a logical issue. If one base codes for 1 amino acid, you can make only 4 amino acids. If 2 bases code for 1 amino acid, like G, A, GU, GC, then you can make 4 times 4, 16 amino acids. That is still not enough to make all 20 amino acids. So the scientists proposed you should have a minimum of 3 letters, G, A, U, or G, A, C, or U, 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 and so on, to make 20 amino acids. So if you make a combination of 4 times 4 times 4, or 4 to the power of 3, 4 cubed is 64 codons. So scientists made all possible sequences of RNA using those 64 codons. That's a lot of work, but it was done in a relatively short time, and they figured out the universal codon information. So that universal codon means universal because from simple bacterium to a complex organism like humans or whale or elephant, they used very similar principle of codon to translation into protein. So once you understand that, the next you need to understand the protein synthesis process. So within protein synthesis process, you'd also think about how the amino acid corresponding to a particular codon is going to be added together. And the amino acid itself, there is no way of communicating with the codon. So tRNA serves as an adapter between the amino acid and the codon in the mRNA. So the tRNA contains something called as anticodon. So the tRNA brings in the right amino acid corresponding to its anticodon that would base pair with the codon. Okay? So the codon in the mRNA, anticodon in the tRNA, that brings the right amino acid. So there's an enzyme called tRNA synthetase that adds the right amino acid to the right tRNA using ATP as the energy source.